Have you ever heard of a species that thrives in toxic waters tainted with pollution and radiation? If not, don't worry, we're the Soulmates and we're here to discuss the Atlantic killifish and how they have evolved to tolerate high toxicity levels within these toxic waters. Now for a brief overview of their diet and habitat. These fish prefer to feed at the water surfaces and their diet consists of phytoplankton, insect larvae, eelgrass, and even their own species eggs. You can find these fish at the Gulf of St. Lawrence in Canada and all the way up the Atlantic coast of North America, in tidal creeks, which is the most ideal environment, saltwater marshes, and estuaries. This information is from Abraham, 1985. Research from McMahon et al. In 2005 analyzed these fish diet and movement patterns by collecting killifish from small salt marshes in Maine and contrasted the gut content and stable isotopes between free-living killifish and those captured for observation. The results show that killifish did not eat plant debris but instead ate locally generated food supplies, even if it meant traveling far distances to obtain them. It also showed that feeding behaviors are similar between killifish that live in small and large marshes.
The killifish, a teleost or ray fin fish, diverged following a whole genome duplication event preserved in their lineage for hundreds of millions of years. Teleosts exhibit diverse sex determination systems which are believed to have facilitated reproductive isolation and speciation within the class. The Atlantic killifish displays phenotypic plasticity, enabling survival in fluctuating estuarine conditions, with varying oxygen, nutrients, temperature, and salinity. Studies indicate that this plasticity is linked to genetic variation in adaptive genes, enhancing the species' ability to thrive in dynamic conditions.
A large population of Atlantic killifish can be found in the contaminated waters of New Bedford Harbor located in Massachusetts. These waters have developed increasingly high toxicity levels because of the exposure to dioxin-like compounds, polychlorinated biphenyls, and other hazardous contaminants linked to the improper disposal of industrial waste and pesticides. These contaminants were determined by a study conducted in 1999 by Nasi et al. In 1999, Nasi and their team performed a study to measure the polychlorinated biphenyl levels within different populations of the Atlantic killifish species. 3 different collection sites were utilized, and it was determined that the Upper Harbor had Atlantic killifish with the most potent PCB levels between the 3 sites. The Atlantic killifish collected from the Upper Harbor had measured PCB concentrations that were as high as 2,100 micrograms. That's 4 orders of magnitude greater than the PCB level guideline that suggests potential adverse biological effects.
Another study by Nasi et al. From 2010 researched the adaptation of the Atlantic killifish to polychlorinated biphenyls, also known as PCBs. This study collected a sample of adult killifish from 24 wild populations and surficial sediments at 24 sites along the North American Atlantic coast. The study measured the surficial sediments to determine the total PCB concentration from each collection site. The map, figure 13, demonstrates where the 24 collection sites were in ascending order of sediment PCB concentration. Sites labeled with stars are stated to have chemically tolerant killifish populations as residents. Table 1 represents all 24 sites and gives quantitative insight into their total PCB concentration per surficial sediments collected and analyzed, as well as other data. This demonstrates that the magnitude of human-mediated pollution affecting the habitats of Atlantic killifish populations is varied.
As the Atlantic killifish inhabit these EPA Superfund sites, which function as landfill and processing plants, they begin to exhibit rapid tolerance to the compounds polycyclic hydrocarbons and polychlorinated biphenyls. These compounds are responsible for cardiac malformation, hemorrhaging, and cranial defects. In 2018, the adult killifish were collected from the New Bedford Harbor on the Atlantic coast, known to be one of thousands contaminated sites at the Superfund status, in Scorton Creek in Massachusetts. Using tissue dissection methods, their PCB levels were tested, and the results shown in the figure were that killifish found in the non-contaminated Scorton Creek had a higher concentration of toxins in their liver than the population at the Superfund site at the harbor. Further research was done to conclude that lower levels of the cytochrome P450-1A and variants AHR1A and AHR2A in the cytochrome ACR signaling pathway of the killifish population in the contaminated sites were the reason for evolution of toxin tolerance.
Hi, I'm going to talk a bit about the future outlook of the Atlantic killifish. The Atlantic killifish are known for their high tolerance to extreme pollution. They show a varying amount of environmental pressures, which include climate change, habitat loss, and coastal pollution. Some of the advantages that we've seen are unique gene expression, which enables resilience to polluted environments, rapid temperature changes, salinity shifts, and varying water quality. So unfortunately, some of those adaptations have limits, and the environmental changes could end up surpassing and outpacing the killifish's ability to adapt and survive. So there's been a focus on conservation, and specifically in protecting coastal ecosystems, monitoring pollution, and implementing broader environmental policies, which are going to be critical for the species' survival.
References. References